Hello, everyone, and welcome back for more human physiology. Last time we started in on chapter 2, and we spent a lot of time talking about water and its capacity to form hydrogen bonds. And we spent quite a lot of time talking about how biologically significant that hydrogen bonding is, whether you're talking about proteins and their stability and the way that they fold, which we'll talk about in the last video for this chapter, DNA molecules and how hydrogen bonding holds two different strands of DNA together, and then most importantly, especially for our coming discussions, the ability of water to act as a biological solvent. So that's going to be kind of at the core of what we talk about in this video, talking about water being the basis for forming solutions and dissolving all sorts of biologically relevant particles and molecules. So let's go ahead and get into this. So first, let's start by defining what a solution is. So a solution is something that you probably heard a lot more about when you took general chemistry, but solutions are very important in biology too, and if for no other reason, because all of the bodily fluids that we talked about in chapter 1, intracellular fluid, the fluid inside of cells, your extracellular fluids, whether you're talking about the blood plasma, the lymph, cerebrospinal fluid, or the interstitial fluid, which is the fluid in between cells, all of those are solutions. And a solution is just a mixture of a liquid solvent and at least one or possibly more dissolved solutes. Now, obviously in biology, your solvent is exclusively going to be water, and we're going to see as we kind of make our way through this discussion just how important water and its capacity to form hydrogen bonds is. So this picture that you're looking at here just kind of gives you a visual representation of how a solution is put together. You've got your solvent. In this case, we are looking at water. And you've got some solute. In this case, the solute's just this unnamed blue solid, this blue crystalline solid. It looks like something that you'd find in a chemistry cabinet. So if we mix the two together, assuming that the solute in this case is miscible in the water, meaning that it can dissolve in the water, we will form a solution in which the solute becomes perfectly integrated into the solvent and they become one homogeneous mixture in that case. Now, biologically speaking, for us in physiology, solutes can be anything that dissolves in the water. It can be glucose molecules, it could be sodium and potassium ions, oxygen gas, although oxygen gas is not very soluble in water, and we'll talk about that later on. But anything that can be dissolved in water is technically going to count as a solute for us. And there are going to be quite a few different solutes that we are going to be tracking throughout the semester, and we will, of course, discuss their relevance in different physiological processes. And like I said, an important thing to keep in mind is that if a particular solute is going to dissolve in water, it has to interact favorably with the solvent, of course, that being water in this case. So before we get too deep into that, we want to go ahead and have a conversation about concentration. So we'll give a formal definition for what concentration is on the next slide. But basically, concentration is going to be your measure of, in a given volume of solution, how much of a particular solute is present. What percentage or what proportion of the solution is taken up by a particular solute. And something that we are going to demonstrate on the next slide is that if the solute concentration in a bodily fluid changes, then the water concentration is going to change as well. So for a little exercise here, take a look at these 5 test tubes here. So what we can assume from the outset is that each of these 4 test tubes has an equal amount of solution. So we'll just say each tube has 100 milliliters of solution. Now, you can just look from the color that each tube has a different chemical composition. They've all got the same volume of solution, but they each have different solute compositions in terms of concentration. So what we're showing here is that we only have one solute. So the solute in this case is just going to be some red solute. Again, we can just assume we took it out of the chemical storage cabinet. So we dissolved different amounts of this red solute in equal amounts of solution. So you can see the leftmost tube, from left to right, we'll just call these tubes A, B, C, and D. Tube A received 10 grams of solute. Tube B received 5 grams, Tube C received 2 grams, and Tube D on the end received only 1 gram. Now, you can use your eyes and you can definitely see that this is true because obviously if our solute is kind of this bright red color, then the tube that received the greatest amount of solute is going to appear the brightest red, and then the tube that received the least amount of it is going to appear less red and kind of a more clearish, kind of pinkish color. Okay, so if we calculate concentration for each of these, again, this is the amount of solute per volume of solution, then working it out so everything comes out in terms of grams per liters, tube A would give us 100 grams per liter, tube B would give us 50 grams per liter, so tube B is half as concentrated as tube A. Tube C would give us 20 grams per liter, so tube C would be 1/5 as concentrated as tube A. And then finally, tube D on the end would give us 10 grams per liter. It is 1/10 as concentrated as tube A. So concentration, as I said, is just a indicator of how much solute is present in a given solution. So that is the amount of solute per volume of solution. So not only does it take into account the mass or number of moles of solute added, it also takes into account the volume as well. It's possible for a small amount of solute to actually produce a very high concentration if your volume is very small as well. Now, here are a couple of questions that we want to ask here, and we need to make sure that we understand this association. Which tube has the highest concentration of solute? Well, we already established that, right? It's tube A on the left. It received 10 grams of solute, and all 4 tubes have the same volume of solution, so it's really a no-brainer that tube A on the left has the highest concentration of solute. But what about the concentration of water? This is kind of an interesting thing to consider because, as we're going to find out in Chapter 3 when we start talking about osmosis, tracking water concentration is not typically something that's done. So it's much more convenient and much more commonly practiced to just keep track of individual solute concentrations. But hopefully you're going to appreciate here pretty soon that being able to infer the relative water concentration from only knowing the solute concentrations, that's going to be an important skill to learn. So what do you think? You may want to pause the video here before I reveal the answer. Which of these tubes do you think has the greatest concentration of water? Well, if you said tube D on the right, you are absolutely correct. All of the tubes have the same volume of solution, but that does not mean they all have the same volume or concentration of water. The idea here is that the more stuff is put into a solution, the lower the percentage of that solution that is made up of water is going to go. So from left to right, tube A has the lowest concentration of water because it has the highest concentration of solute. Tube D on the right has the highest concentration of water because it has the lowest concentration of solute. If you consider a fifth tube, we'll call it tube E, that literally all it contains is 100 milliliters of water, then that would literally just be 100% water, right? It's not possible for us to have something that's more than 100% in that case. Tube D, the 1 gram per 100 milliliters there on the right there, that is not 100% water, but it's maybe somewhere in the neighborhood of like 90% water. Tube A on the very left, that is a much lower proportion of water because a much higher percentage of that solution is taken up with solute. So this will be a kind of a concept that we review again in Chapter 3 when we introduce osmosis, but I want you to go ahead and try your best to understand that association. The more stuff you dissolve in solution, the lower the relative water concentration is going to go. Okay, and then another thing that is going to become important later on, I do want to go ahead and introduce it now just so you can kind of go ahead and get this into your head. The reason why we want to keep track of individual solute concentrations, especially when it comes to human physiology, is because all of your bodily fluids, intracellular fluid, interstitial fluid, blood plasma, they're in a state of what we call chemical disequilibrium, meaning that if your body is doing its job correctly, there never, ever, ever should be an equal concentration of solutes on either side of a membrane or in between body compartments. So if you look at the breakdown here in terms of individual concentrations, in terms of millimoles per liter, of key solutes like sodium, potassium, chloride, bicarbonate, and individual proteins, you can see by comparing different bodily fluids to one another that none of these solutes are really equal to each other in most cases. The important ones to really pay attention to here are going to be sodium and potassium, and we will definitely need to keep track of this in the future. So if you look at sodium, well, sodium, there appears to be a very low concentration of sodium in intracellular fluid, but a very high concentration of sodium in the extracellular fluids. Well, what about potassium? Well, for potassium, it is just the opposite. Potassium is at a very high concentration in the intracellular fluid and at very low concentrations in the extracellular fluids. Being able to appreciate that's going to be really important for when we eventually start talking about the nervous system and the ability of nerves to send electrical signals. We'll find out that sodium and potassium are key players in those processes. But for now, just make sure you understand that individual solutes are maintained at very different concentrations between different body compartments, and there is a very good reason for making sure that that does not change. Okay, so let's talk about individual solutes. So we already know that our solvent in most cases, really in all cases for biology, it's going to be water. So we know what our solvent is going to be. Well, what about individual solutes? So we already said that a solute is just any molecule or atom or ion or whatever that can be dissolved in water. Well, water, we've already established in the last video, is a very polar substance. So water needs to be able to form hydrogen bonds with any solutes that are going to become dissolved. So we need to be able to categorize solutes on the basis of whether they will interact favorably with water or whether they will interact unfavorably with water. So let's go ahead and introduce a couple of new terms here. The first term is called hydrophilic. If a solute is hydrophilic, this is a solute that is going to very readily interact with water, and these are basically going to be your solutes that are very, very soluble in water. The word hydrophilic has two roots to it. Hydro, of course, means water, and the word phile obviously means lover of. If you're a bibliophile, you love books. If you're an audiophile, you're a great appreciator of music or sound. So aphilia refers to a great appreciation or a great love for something. So if a solute is very hydrophilic, it has a great and deep love and affinity for water. So these are going to be your solutes that are very, very, very soluble in water, either because they are very polar themselves or because they have some sort of electrical charge. What they have in common is that these are solutes that water can form hydrogen bonds with. Examples of hydrophilic solutes that we will see throughout the semester are salts, sugars, alcohols, and globular proteins. Now, in the case of salts, which are going to break up into individual positive and negative charges, it's not hard at this point for you to see and understand why water would be able to dissolve them. The partial negatives and partial positives would be able to interact with those corresponding negative and positive charges with the ions. But for sugars, alcohols, and proteins, you'll be able to appreciate why these are very soluble in water later on when we start talking about functional groups and organic molecules. So just hang tight until we get to that particular point. Other molecules which are not going to interact favorably with water are called hydrophobic solutes. The word hydrophobic has, again, important root meanings here. Hydro, again, means water, but this time the word phobia refers to a fear of something. So it would not be unreasonable to say that a hydrophobic solute has a fear of water. And what do you do when you are afraid of something? Well, you try to do anything and everything in your power to make sure that you never come into contact with the thing that you're afraid of. So hydrophobic solutes are going to try their best to stay away from water. So these are going to be the types of solutes that water cannot form hydrogen bonds with. So they're going to be nonpolar and electrically neutral. So basically there's nothing for water to form hydrogen bonds with, and therefore water and these hydrophobic solutes are going to have a very, very, very tough time interacting with one another. Your best examples of these are going to be gases like oxygen and carbon dioxide and nitrogen. Gases, by their nature, tend to be very small and nonpolar and have no electrical charge. Lipids, like fats and steroids, are very hydrophobic, as you're going to find out in your on your own work in this chapter when you look at lipids. And then particular types of proteins called fibrous proteins are also very hydrophobic as well. The distinction between globular proteins and fibrous proteins is something that we will discuss at the very end of this chapter. So let's look at particular examples here to see with our own eyes just how some of these solutes either do or do not interact favorably with water. Let's start with some of our hydrophilic solutes. Let's start with salts. So a salt is just an ionic compound like sodium chloride or potassium iodide or magnesium sulfate or whatever you want to come up with. So again, you've got on your own work to kind of catch yourselves up on how ionic bonding works, but salts are going to be very hydrophilic solutes. So in something like table salt, sodium chloride, if you just have a sample of table salt as its crystalline solid form, the ionic bonding, those electrostatic interactions holding the positively charged sodium cations and the negatively charged chloride anions together are going to be fairly strong. So that's what holds it together as a crystalline solid. If you throw some salt into a glass of water, water is going to break up those ionic bonds so that the salt is going to dissolve. And now, instead of having intact sodium chloride units, you are going to break them apart so that you no longer have any intact sodium chloride. What you're going to have now is a solution that contains sodium ions and chloride ions. So right now, if you take a beaker of pure water and you throw salt in it, you are now going to have a solution, after everything's dissolved, of course, you are now going to have a solution that has two different solutes in equal concentrations. You're going to have an equal concentration of sodium ions and an equal concentration of chloride ions. Both of those solutes will be kept separate from one another, so the table salt goes off by the wayside. You no longer have intact sodium chloride, you've just got sodium and you've just got chloride. The pictures here also paint a kind of a representation of how water is able to do this. Sodium, on the one hand, of course, carries a +1 electrical charge, so the partially negative oxygen atoms of individual water molecules are able to surround and solvate those sodium ions. And then similarly with a chloride ion, which carries a -1 electrical charge, the partially positive hydrogen atoms from individual water molecules are able to surround and solvate chloride ions as well. So the idea here, as we said, is that salts will completely dissociate in water. So like I said, there's not going to any longer be any intact sodium chloride. You're just going to have a solution that has individual sodium ions and individual chloride ions. And I've already kind of given you a sneak peek of why sodium and potassium are going to be very important. They form the basis for how electrical currents are conducted along the lengths of neuron axons in nervous system signaling. So being able to appreciate how an electrically charged ion can exist by itself in an aqueous solution, that's going to be kind of important later on. Okay, so those are examples of solutes that carry full electrical charges. They're hydrophilic. What about polar solutes? Well, polar solutes will also dissolve in water as well. They are also hydrophilic. So water is polar, and polar solutes like sugars and alcohols are also polar as well. You're going to find out pretty quickly here that like dissolves like. Polar molecules want to dissolve in a polar solvent. So if you look at something like glucose here, again, we're going to learn more about functional groups and how functional groups can be used as a predictor of solubility in water later on in the chapter. But for now, all I want you to appreciate is that this molecule of glucose has a lot of oxygen atoms. So we've got a lot of oxygen atoms here. So a lot of these bonds between oxygen and hydrogen, oxygen and hydrogen, oxygen and hydrogen, the same rule is going to apply here as what we saw with water earlier. The oxygen atoms are more electronegative than the hydrogen atoms, so you're going to have polar covalent bonds here, and that is going to form basically something that water can grab onto via hydrogen bonding. So you can see here that this sugar molecule gets completely surrounded by water, and that is due in large part to the ability of water to form hydrogen bonds between the electronegative oxygen of the water molecule and the partially positive hydrogen atom of each hydroxyl functional group that surrounds the individual sugar molecule. So just as the case was with salts, hydrogen bonding forms the basis for how these polar molecules can become dissolved in water. And the same thing would apply to alcohols as well. Alcohols have the exact same functional groups that sugars have, just a lot less of them. Okay, so now let's take a look at a hydrophobic solute. So trying to dissolve a non-polar hydrophobic solute in a polar solvent like water. Well, before we dive into that, I've got a question for you. Have you ever heard the expression, oil and water do not mix? Well, whether you've heard this expression or not, you've likely seen this phenomenon in effect if you've ever tried to make homemade salad dressing. What happens when you make homemade salad dressing and then you leave it out for a little bit? Well, the watery component and the oil component separate from each other, right? So if you want to get all those flavors mixed together again, you got to shake up the bottle before you pour it onto your salad. Well, the question here, though, is that why not? Why do oil and water not mix together? Well, this picture here should kind of help to illustrate what we're talking about here. Water, we're already familiar with the idea that these individual water molecules are very polar. They form hydrogen bonds with one another. And if those water molecules are going to dissolve any potential solutes, the water molecules need to be able to hydrogen bond with those respective solute molecules. Well, look at the oil molecules here. So oils, as you're going to find out in your on your own segment this week, oils tend to have a lot of carbon and hydrogen. Well, remember what we said about carbon and hydrogen in the previous video. Carbon and hydrogen have roughly equivalent electronegativities. So an oil molecule like what you're seeing here is pretty much going to be nothing but non-polar covalent bonds. That means that there is nothing for water to grab onto. Water cannot hydrogen bond with any of the atoms within that oil molecule. So what is going to happen instead is that water is going to reject the oil and that is going to cause the oil and the water to separate into two phases. The oil sits on top because it has a lower density, and then the water is on bottom because it has a higher density. We try our best to minimize the interaction between the hydrophobic molecule and the water itself. Like I said, a hydrophobic molecule is going to do its best to get away from water, and that is what oil is trying to do here. So ultimately, the reason why oil and water do not mix is because there is a lack of stabilizing forces. We cannot form any hydrogen bonds, so the oil molecules will sequester themselves as far away from the water as they possibly can. And there is actually a formal and very important name for this type of interaction. This is called a hydrophobic interaction. And this is something that goes beyond just trying to mix oil and water together. Hydrophobic interactions form the basis for a lot of important molecular processes, and we are going to see an important example of that later on when we talk about how proteins fold. If you've got a component to something, whether it's an individual molecule or a part of a molecule that does not like water, it's in your best interest to try to satisfy its desire to be as far away from water as possible. Now, hopefully what you've taken away from all this is listed here. Polar solutes will dissolve in polar solvents, and nonpolar solutes will dissolve in nonpolar solvents. Like dissolves like. All right, that is going to do it for this video. So join us next time and we will kind of continue on our discussion about how important water is, and we will talk about the concept of pH and buffers in physiological settings. So thank you for your attention, and I will see you next time.